Have you ever visited Japan and sat down on one of their high-tech toilet bowls? You know, one of those toilet bowls with lots of buttons and curious but strange functions which no one has ever heard of? And then you think to yourself, how does this thing work? That might just be the same thing that you think as you look at the kidney tubule. Because it's made out of so many parts and it's surrounded by blood vessels, and you're just like, how does this thing work? Well, fear not. That is the question we're going to explore in this video. In this video, we're going to take a look at how urine is formed by the kidney tubule and how blood is filtered by the kidney tubule. Now, urine is formed through two main processes. The first one is called ultrafiltration, and the second one, selective reabsorption. And we're going to take a closer look at each of these processes taking place in a kidney tubule. So by the end of this video, you should be able to have an understanding of how urine is formed and what is ultrafiltration and what is selective reabsorption. Now, the textbook pages are as follows, so have a read of those as well as you go along. Alright, let's start off by looking at ultrafiltration. So the formation of urine starts with this process called ultrafiltration. And ultrafiltration takes place in the Bowman's capsule, at that cup-like structure at the start of the kidney tubule. So what is ultrafiltration? Well, it is a process where small molecules are removed from the blood or filtered out from the blood. So here's a diagram showing us what happens during ultrafiltration. And what you see here is the Bowman's capsule and the glomerulus, right, which is the mass of blood capillaries found inside the Bowman's capsule. So what happens during ultrafiltration? Well, blood enters the glomerulus at a very high blood pressure, a very high hydrostatic blood pressure. Hydrostatic just means something to do with a liquid. Right, so there's a very high pressure and the blood rushes in at that high pressure. And what that high pressure does is that it forces out water and other small molecules from the glomerulus. So water and small molecules are forced out by the high pressure, forced out of the glomerulus into the Bowman's capsule. And it forms a filtrate. Right? It's a bit like filtration, right, in chemistry, which you learn. So water and small molecules will pass out of the glomerulus and form a filtrate in the Bowman's capsule. So what substances remain in the glomerulus and what substances are filtered out, pass out of the glomerulus and form the filtrate? Well, the substances which remain in the glomerulus are those substances which are relatively large. For example, your red blood cells, your white blood cells, your platelets, they all remain inside the glomerulus. And large molecules, such as large proteins and fats, these will also remain in your glomerulus. So large proteins, for example, enzymes, or you'll learn about it soon, hormones, these will remain inside the glomerulus. Now, what gets forced out of the glomerulus are smaller molecules. For example, water gets forced out, all your glucose will also get forced out, amino acids will get forced out, mineral salts will get forced out, and your nitrogenous waste products like urea will also get forced out. Now, of course, some water does remain inside the blood vessel. Otherwise, there wouldn't be any liquid left in your blood. So a lot of the water gets forced out, but some will remain in the glomerulus as well. Now, the glomerulus and the Bowman's capsule are very well designed for ultrafiltration. They are very well adapted for their function. So, for example, in ultrafiltration, we need a very high blood pressure in the glomerulus. Now, how is this high pressure created? It's something to do with the structure of the glomerulus. In this diagram, you see that the blood enters the glomerulus through the afferent arteriole, and blood leaves the glomerulus through the efferent arteriole. Right? How you remember that is that You know, A is the alphabet that comes first, then E, right? So afferent arteriole is the blood moving into the glomerulus, and efferent is the blood moving out. Interestingly, if you look at this diagram, what do you observe? You'll see a difference in the diameter of the arterioles. Now, the afferent arteriole that brings blood to the glomerulus has a larger diameter than the efferent arteriole that carries blood away. And it is this structure that causes a very high pressure in the glomerulus. Why? Now that you just pause the video for a while and think about it, why is it that if this diameter is small and this one is large, that there's a very high pressure here? Just think about it for a while. Well, if you're still sitting there wondering to yourself, here's a simple example that will hopefully help you get the answer. So imagine that you have a garden hose and it has some holes inside it. So it's a holy hose, and water can come out through the holes in the hose. Well, what happens if we squeeze the hose on this end and we decrease the diameter here? What will happen to the water spurting or squirting out the hose? Well, hopefully your answer is that the pressure here increases, right? Because water, in a sense, it can't move through this part so easily. So there's a buildup of pressure here, and you can see that the water will squirt out more violently and with greater force. It's very similar in the glomerulus, where the afferent arteriole has a larger diameter than the efferent arteriole, and so the pressure in the glomerulus is very high, which helps ultrafiltration. Alright, so now we know how this high pressure in the glomerulus is formed. But the next question is, how does the filtering occur? What is the filter here in the glomerulus? How is a filter formed here in the glomerulus? Well, let's take a look at that now. So what forms the filter? Well, it turns out that the glomerulus capillary wall, or the glomerular capillary wall, has many small pores. So here is a diagram of the glomerulus. This is the blood capillary, the glomerulus, and the capillary wall has many small pores, as you can see. Now, furthermore, the glomerular capillary, the glomerulus, is surrounded by a basement membrane with very small pores, which you can't even see. And these pores act as filters during ultrafiltration, right? So what you get is a very high pressure in the glomerulus, and then the glomerular capillary wall, which I'm highlighting for you here, the wall of the capillaries have very small pores, and they're surrounded by a basement membrane with even smaller pores. So there is a filter here. So the pressure pushes substances through the filter. And as we have seen, the larger substances remain in the glomerulus while the smaller ones get filtered out to form a filtrate in the Bowman's capsule. So that, in a nutshell, is ultrafiltration. And we have seen how the glomerulus is adapted for ultrafiltration, how the high pressure is created, and what forms the filter. Alright, so now that we've looked at ultrafiltration, we're going to move on to the second stage in urine formation, which is selective reabsorption.